This is Lecture 5 in BBSP 710. As the title implies, today we're going to give a brief introduction to R and also an explanation of how you can do some simple sampling operations in R. So what is R? It's an open source statistical computing package, and it has a number of advantages for a bench scientist looking to do statistical analysis. First of all, it's free, and it can be installed on Windows, Mac, Linux, whatever platform you want to install it on. No need to worry about licensing or going to the computer lab or whatever else. You can just download it on your computer and start using it. And it's probably, I may be biased, but in my opinion, the most powerful statistical package out there. I use R myself for my own research because it's simply the best platform there is if you need to do programming, things like that. And it's become increasingly popular in biomedical research applications. The Bioconductor project, a lot of the tools for analyzing high throughput biological data are all written in R, so knowing how to use R, you can use these tools. The downside to R is that it's not very intuitive and it can be difficult to use. So I'll post some materials with the general introduction to R on Sakai, and you're also welcome to email me any questions that you might have. Well, at any rate, to download R, you go to the URL that I've listed on this slide. Hopefully it's fairly self-explanatory. As you can see, you just select what type of operating system you're using. Linux, Mac, or Windows, then just download and click on it. Most people don't seem to have problems, but if you do have problems, you can send me an email, and we'll also be going over this in class if necessary. To start R, you can double-click on the R icon, or if you're a geek like me that likes to use the command line in Linux or Mac, you can just type capital letter R on the command line. One way or another, you'll end up with a screen that looks something like the screen capture that I have here. And one of the first things that you'll need to do in R is setting the working directory. This is probably the single biggest problem people have when they try to do the second homework assignment, quite honestly. The way R works, it stores all the variables you create and all the data sets that you read in a file called .rdata in whatever directory R is running. And it's generally a good idea to save each project or each homework assignment or whatever in a separate directory. And to change the directory in Windows, it's under the File menu. In Mac, I believe you have to go up to the menu at the top that just says Miss or Miscellaneous. In either case, then you click on Change Directory. And you can also do this from the command line if you want to change your directory manually. Before you try to read in data, make sure that you've changed your directory to the folder where the data is stored. Otherwise, R won't be able to find it, or you won't be able to read it in to do your homework. Oh, now I'm going to briefly describe how to get help in R. For any command in R, if you type question mark and then the name of the command, it'll give you detailed information about the command. Say I can't remember how to use the setwd command, which is used to set the working directory that I just described. You can type question mark setwd, and it will open up a little help file like this, where it explains how to set the working directory. And if you can't remember the name of a command, you can use a command called help.search. In this case, say I want to change the directory, but I can't remember the command that's used to change the working directory. So I say help.search, and then in quotation marks I say directory, and it lists the various help files that are associated with commands. I look, say, aha, get wd, get or set the working directory. I could then type question mark get wd to get the detailed help with all the commands and so forth. And so if you want to quit R, the command is you go Q and then in parentheses and then in quotation marks no. If you don't want to save your work, then you say yes in quotation marks. If you do want to save your work, if you're using Windows or Mac, you can also quit using a drop-down menu. Just go File, Quit, and it'll ask you whether or not you want to save your work. Important note is that R is case sensitive, so that if you try to quit by saying capital Q and then yes, or if you say set WD in caps, that won't work. You have to do the same case, either uppercase or lowercase, exactly as these commands appear on the slides. Oh, here's an example of reading data into R. This is a data set that's built into R, although I'll post it on Sakai so that you can practice doing this yourself. This is a data set on plant growth. In this particular experiment, there are 30 plants with 2 possible treatments. 10 plants got treatment 1, 10 plants got treatment 2, and then there's also 10 controls. We want to compare the dry weights of the plants in these 3 different groups. And it looks like part of my slide got cut off again, unfortunately. That's supposed to say control treatment 1, treatment 2. One way or another, the raw data looks like this. And say you have this data set and you want to load it into R to manipulate it. So one thing that you can do is to input your data manually. And say I create a vector called weight that consists of all the numbers that you saw on the previous slide. And then I create a second vector called group, which contains, and these are commands I'll explain in more detail over the course of the next couple slides, but what this says is take a group called control, repeat it 10 times, take a group called treatment 1, repeat it 10 times, then take a group called treatment 2 and repeat it 10 times. I end up with a vector of length 30, and the vector of length 30 is just the weights that I showed you on the previous slide. There's the date again, and as you can see, I've entered that same data into R, and I've also created a vector called group that has control repeated 10 times because the first 10 plants are controls, then the treatment 1 plants repeated 10 times, and the treatment 2 plants repeated 10 times. Oh, let me explain what these various commands in R mean. That the C command in R concatenates its arguments to create a vector, and you can use this little arrow operator to assign a value to a variable. You can also use an equal sign. In older versions of R, the equal sign didn't work. You had to use the little arrow thing. I've got into the habit of using the arrow, so that's what you'll see on most of my slides, but an equal sign works equally well. So if I wanted to create a vector called blah that contained the elements 3, 6, 4, 6, and 9, I say blah, arrow, 3, 6, 4, 6, 9, you can see I've created a vector called blah. If I wanted to create a vector containing the names of the students in my old Bio 610 class, I could say Bio 610 equals and then a vector of names. R is perfectly happy with character vectors in addition to numeric vectors, although in practice you'll normally want to use numeric vectors. And if you want to see all of the currently defined variables in your workspace, you can use the objects command. I say objects and it gives me the BIOS 610 vector I just created, the blah vector I just created, and also these weight and group variables that I defined a few slides back from the plant growth data set. If I want to remove a variable From R, I can use the rm command, say I'm not going to use this variable called blah anymore, so I want to get rid of it. So I say remove blah, and then I run the objects command again. You see blah is no longer on the list of variables. I nuked it by using the remove command. And if you want to save your current workspace, you can type the command save.image, open and close parentheses. That will save all the data in your workspace to your hard drive. It's a good idea to do periodically just in case R crashes or your computer dies or whatever the case may be, so that you don't lose all your work. The next R command is this command called rep, which tells R to create a vector with the first argument repeated a certain number of times. If I wanted to create a vector containing the number 3 repeated 5 times, I could say rep 3, 5, we get 5 3s. Or if I say rep 4, 10, I get a total of 10 4s. And if I say C rep 3, 5, 4, 10, that creates a vector of length 3, a vector of length 15 consisting of 3 5s and 4 10s, one after the other. C, as I said before, concatenates two vectors together, so it takes the two vectors that you saw previously, puts them into one big vector. And the factor command in R tells R to treat a specified vector as categorical data. That if I say factor C1, 1, 2, 2, 3, rather than treating it as number 1, number 2, number 3, it treats it as a categorical variable with 3 categories: category 1, category 2, category 3. The categories don't have to be numbers. You could have, say, category red, category white, category green. A factor in R is a categorical variable as opposed to a numeric variable. If you need to import data from Excel into R, which you'll probably want to do quite regularly, both in this class and outside the class if you ever use R for your actual work, you need to go through a couple steps to do this. To read data into R, first of all, you open the data in Excel, and then you have to save it as file type text CSV. And you can accept the default field /text delimiters, and it'll create a CSV file containing the spreadsheet that you wanted to read into R. And then to read the data into R, I can say like plant growth equals read.csv plant growth.csv. This plant growth data set contains 2 columns. I'll put a copy of it on Sakai so you can try this yourself if you want. One column called weights, which is the list of the 30 weights. Another column called group has the different groups: control, treatment 1, and treatment 2. And in R, when you use the read.csv command, it creates a data structure called a data frame. You can think of a data frame as just basically an Excel spreadsheet, and to access a specific column of your data frame, you use this $sign. So here you see plant growth $weight, plant growth $group. That's saying, when you say plant growth $weight, that tells R that you want to use the column of this data frame, the column of this spreadsheet called weight, and plant growth $group is the column called group. Um, in this case, the spreadsheet contains a header at the top. The first row of the spreadsheet will say weight and then group, just sort of the column header, so that R knows what they're called. One important thing, before you use read.csv, make sure you change the working data, or make sure you change the working directory to the directory where you saved your data set. If you didn't change the working directory, R won't be able to find it and you'll get an error when you run this command. And so now I'll give an example of doing randomization in R that in last week's lectures, I talked about the importance of randomization in experimental design, that we should randomly assign experimental units to treatment groups and control groups, and that's better than any sort of non-random selection. For small studies, this is relatively straightforward. You can just flip a coin, roll dice, whatever the case may be. But if you have a study with like 1,000 participants, you'll probably want to use a computer to do your randomization. So to give a simple example of this, let's say you just have a simple case control design. We want to test a certain treatment on a cohort of 20 rats. 10 rats will receive the treatment. The other 10 rats will receive a placebo. We'll assume this population of rats is homogeneous, so we don't need to do any blocking. You can do this in R with the sample command. Here's an explanation of the sample command. If you type $sample, it'll give you this message, but I'll explain it more by example over the course of the next several slides. Say you've just numbered your rats 1 through 20. You can use the following command in R to randomize. You say sample 1:20, 10. And 1:20, that'll just give you the numbers 1, 2, 3, 4, 5, 6, 7, up to 20. I'll describe this in more detail on the next slide. And the 10 says choose a sample of size 10. So this gives you the IDs of the 10 rats that you would include in your treatment group. For example, 11, 6, 3, 4, 10, 13, 15, 14, 5, 12. You would put those in your treatment group, the remaining in your control group. Hopefully that's simple enough. And in R, if you say 1:5, that just gives you the numbers 1, 2, 3, 4, 5. As I said before, it means exactly the same thing as 1, 2, 3, 4, 5. As you can see on this slide below, if you did like 1 to 100 or 1 to 1,000, you don't want to type the whole thing. So being able to say 1:100 to give you the numbers 1 to 100 is convenient sometimes. R can also randomize objects that are not numeric. Say I want to randomly choose some students from my old class to pick on, but I don't want to be accused of racial or gender discrimination, so I choose my victims randomly. And this example is kind of a joke, but when I create the discussion groups to discuss the problems in BBSP 710, I actually do some randomization in R, just like I describe here. Let's say I want to pick 4 students from my old Bio 610 class to pick on. I sample from the Bio 610 vector that I created earlier. I end up with a sample of size for the 4 students that I'm going to be mean to. Now let's take the plant growth example that I described earlier. Let's say that I have 30 plants and 2 different treatments, and 10 plants are going to be assigned to treatment 1, 10 plants are assigned to treatment 2, and the final 10 plants are controls. Well, I can do that in R like this, that if I say sample 1:30, that if I don't give a sample size, it just permutes the vector 1 to 30, takes the numbers 1 to 30 and puts them in random order. So in this case, what I'll say is I'll just scramble the order of 1 to 30, then I'll take the first 10, put them in treatment 1, the second 10, put them in treatment 2, and the final 10 and put them in treatment 3. So I scramble all the numbers 1 through 30, it looks like this. Then the first 10, I see here 4, 2, 22, 9, 15, etc. They get treatment 1. The group that ended up in 19, 14, 3, 27 gets treatment 2. the group that ended up in 26, 6, 11, 5, etc., those are the controls. Just a quick syntax note. In general, in R, if you use those square brackets like I did on the previous slide, that will give you a subset of a vector. So if you remember my Bio 610 class list that I used previously, if I want the fifth one on the list, which in this case is Jennifer, I can go Bio 610 of 5, I get Jennifer. If I want the fifth through eighth people on the list, I go Bio 610 of 5 colon 8. I get Jennifer, Catherine, Ankunda, Alicia. The people numbered 5 through 8. In general, if you have a vector in R and use these square brackets and say 1 colon 5, that gives you the first 5, whatever the case may be. So now let's remember the turkey growth example. Show how you could do this type of randomization in R. If you recall this example from last week, methionines added to turkeys to enhance their growth. We want to compare 3 different types of methionine that we call T1, T2, and T3, and we'll measure the weights of the 12 turkeys over a 3-week period. So we stack the cages on top of one another 4 layers high. There's 3 cages on the floor, 3 cages on the second row, 3 cages on the third row, etc. The cages near the ceiling are warmer than the cages near the floor, and the amount the turkey eats depends on the temperature. So here we use blocking, that on each level we assign 1 turkey to each treatment group. So we basically do the randomization separately on each level of the cages. Well, the simplest way to do this in R is just randomize 4 times. So you can say, so we say sample 1 colon 3 in R and we get 3, 2, 1. So we can say the first turkey gets T3, the second turkey gets T2, the third turkey gets T1 on the bottom row. Then on the second row, the first turkey gets T3, the second turkey gets T1, the third turkey gets T2, and so on down the line. You can simply do this 4 times and randomize separately within each block. There's more elegant ways to do this, but for the purpose of this class, this works, and it's hopefully less confusing. Well, our commands to remember for today, if you say question mark followed by the command, it'll display a help file for a command. If you do help.search, it'll search for an appropriate keyword. The C command will concatenate the arguments into a single vector. The rep command will repeat a given number or any object a certain number of times. If you say objects, that'll list the variables you've defined in your current workspace. RM is to remove a variable. Save.image saves your current workspace. Read.csv loads data from a CSV file. 1 colon 5 is shorthand for the numbers 1 up to 5. And if you use these square brackets, you can return an a given element of a vector, and if you say sample 1 colon 10 comma 3, that'll give you a sample size 3 from numbers 1 to 10. You can use this for sampling in general.